I'd now like to say a few words about Debbie Marks, our second award winner. Debbie, thanks for coming on down. So again, I'd like to just offer a few quotes from the letters that were a part of her nomination package. Galit Lahav, who nominated Debbie for this award, writes, "Dr. Marks' work integrates exceptional creativity in the computational analysis of large-scale genomic data with a rigorous understanding of biochemistry and biophysics, and a rare, remarkable instinct for how computation can be used to answer key biological questions. Importantly, Dr. Marks does not simply use general approaches from computer science to analyze existing biological data, but rather develops novel computational approaches specifically designed to gain insight into central biological problems." Edward Marcotte wrote that her work on evolutionary coupling was a seminal breakthrough. For more than 20 years, biologists had known about the latent information in protein amino acid sequences. Dr. Marks's contribution of the appropriate mathematical framework, maximum entropy, did what others had failed to do, suddenly opened the ability to compute protein 3D structures with more power than ever. Aviv Regev wrote that Debbie is a foremost leader in her field, developing new methods for advancing our understanding of protein structure and functions through mathematical methods and AI models and AI. She has repeatedly made crucial, groundbreaking, and sophisticated contributions to computational and structural biology that have driven dramatic advances, including the foundational work on predicting protein structure solely from the sequence, on which methods like AlphaFold and RosettaFold then followed. Aviv and fellow letter writers Olga Troyanskaya and Madan Babu also point out that her more recent contributions to building deep generative models for predicting pathogenic effects of missense variants. They said applying this to over 3,000 genes in the human genome revealed an accuracy equal to or better than traditional high-throughput experimental assays. She's now working in collaboration with clinical genetics groups throughout the world to take her methods into the clinic. I'd also like to just point out that I think this work highlights the importance of academic research and its interplay with industrial research in the general field of scientific AI. And I'll also mention that Debbie pointed out to me that she started much of her scientific investigations a little later than most people, and thinks of this as a great, hopefully, inspiration for many people to consider that it's never too late to embark on new directions and new careers. Please join me in welcoming and congratulating Debbie again.
Hi, guys. Right, where do I go from there and that amazing talk previously? There is a flicker here. It looks like it's this one. Okay. Oh, yeah. So the reason I put up the second slide, which you'll see in a minute, you haven't yet seen, is because otherwise I will go so slowly or have too many slides that I won't be able to thank everybody. And the first thing I want to do before I thank the people on the next slide is just to say, amazingly honored. I find it really hard to not be sarcastic. I'm English, but I'm not being sarcastic. To be in this room and be awarded the prize today, because much as I'm not self-deprecating, I also like to think I know my place, and I'm very honored and really want to take this opportunity really to motivate you guys in the room and your labs or your friends to go out there and experiment on a computer with biology. Okay. So here's the thanks to my lab, which is at the beginning and the end, and the funders. I suppose I should cross off the NIH, but maybe we'll get it back. Actually, there should be another couple on there that are government funded that I lost. But I'm not complaining. I am super lucky. So from a very young age, and I promise it's not all about biomedicine, I'm very much with Francis in the sense of I want to solve everything in the world using biological life and programming biology. But this is one example is, you know, for a long time, genetics has been about the changes in your DNA, and then does it give you red hair or a red skirt or propensity to diabetes or whatever it is. And, you know, there's a massive industry and billions of dollars in all the intermediate phenotypes that we know and love in biology, like the ones you will have heard of, like protein structure, which actually are quite near phenotypes if you think about it from the genotype, and are way off how cells will be in a body for human genetics or drug discovery, just saying. And then the other arrow, which of course we all know, is, especially as I'm addicted to generative models, which we can talk about in a minute, is the ability of them to design. Now, people hear the word generative who are outside the field that you're all in, and they think, well, what's that got to do with prediction? They actually think we should still have heuristics. And I think one of the things that I've realized in the past 6 or 7 years, I've had my lab for 10 years, PhD, I don't know, 12 years ago, something like that. And I realized that it's a sort of, you know, there's a lot of sexiness around design, which we have to be able to do, and we need to find methods for it, and it has enormous potential. But actually, so does prediction, but it seems a bit old-fashioned, so I'm going to cover that. Okay, so the theme, generative AI models decode and encode the language of life. Language of life not being the words we speak, but the DNA. And I'm going to talk about 3 things. So I'm going to give you like 3 vignettes of what we've been doing in the lab and in the last 10 years. And really, I suppose the mission has been to say, what are really the unmet challenges in biology writ large? I don't mean I want a drug for my disease tomorrow, because perhaps that's too bold. But what are the challenges that down the tree that would open up a field? And I don't, you know, because I was already cynical, I didn't want to just get my AUC better than their AUC or whatever. I encouraged the lab to think about what the goals would be. And so these are just 3 examples of where we went with that. And one of the things that comes up all the time with students especially is, you know, given the feeding frenzy over design models for proteins at the moment, you know, that got hugely advanced by the discovery of AlphaFold, because, you know, then people all wanted to do the same thing. So is like, which generative kind of model should I use? Well, I don't know. Do you? It depends what you want to do. And that's what we found over and over again, actually, which is the combinations of models that you have and this so-called fine-tuning, which actually is often the main bit of it, by the way, actually matters hugely. And then, of course, the data. So there, I may disagree with this morning's talk, but totally loved her, which was, you know, physics-based. So I kind of, you know, I'd love to work everything out in biology from basic physics, but it's a bridge too far for me. I'm just stupid. And so, I know, I'm just... People talk about, though, learning basic things about molecular interactions. So let's do it by molecular biology. We know each interaction, we know the residues, we know the stoichiometry, Let's work out how they interact. And then let's look at complexes, and then let's look at dynamics, and then let's work out what's in a cell. And then let's work out what cells interact with what. Oh my God, I'm looking at the 23rd century at that point, in my view. And based on not necessarily knowing what the things are at each scale. So I'm a little bit more expedient than that. And I do think that we have to have a question and a goal. So one of the things, and I just put this up because otherwise I didn't know where to put it in the slide deck, because we put it out this week. It's not the main thing we do, but just to illustrate how we don't care what models people use for the problems we want to solve, right? Which is, I want to design a protein that works at this pH with this efficiency. And I might do it, I want it in a 96-well plate. I only want to test 10 to get 1, or I only want to test 10,000 in a library. to get one. And we realized that there are all these models, and so we're making a sort of open source thing where you can design your own, and you get out of it the things that you can send to the twist or wherever, TenScript. You don't get out the actual sequences. That also came up this morning. I wanted to just tell you about that resource, because part of what I do in the lab, and it's hard as an academic to do that, is to create a resource that's genuinely useful, because, you know, there's masses of billions of dollars now put into antibody design, as we all know, biologics, and lots of companies doing it. But actually, the different methods in the field are very siloed, and whoever got the latest recently, it's hard to know how to discriminate. So we wanted to create something for researchers and bench scientists. Okay, what are generative models? I don't really have to tell you that. Actually, I don't know what they are. Does anybody know what generative models are? I think it's kind of a word for probabilistic, basically. And yes, guess what? Biology tends to be full of probabilistic relationships between entities, molecular or cellular, rather than specificity, which is what we were all after, because we imagined lock and key, right? Like the ligand going into the and it ain't like that. So how do we redesign things that we want to work in certain circumstances, either to exploit the probabilistic framework or to actually design against it and be more specific? And the other things that I find with biologists that I talk to is that they don't realize that generative models are also extremely good, potentially, at predicting what's feasible, right? So therefore, they're good at predicting. And there's, just for the young people in the room, any left, and the old people in the room who want to do new science, or the same science with this in mind, there are many challenges in this area. So you might feel that, you know, people have got these Nobel Prizes, or there's a paper every 3 days. Many of these challenges are just not solved. And if anybody wants to talk to me about them, happy to do that. So where do we start in my lab? I only started my lab about 11 years. I used to say 10 years, and now it's 11 years ago. And it was an accident. I was sorted from mathematics and thought I'd stay in sort of cryptography or something with no value judgments involved, and realized that my starting love was, you know, evolution. So, you know, evolutionary sequences, it turns out, are the result of billions of experiments. So can't we use it? Well, we all know we can, right? We've known for 60 years that we can, since we first started sequencing. So it's one of those things where sequencing the genome and genes has kind of crept up on us. Now it's like easy peasy lemon squeezy, sheepy weepy. And people don't realize, actually, I worked in collaboration with people doing antibody discovery for very important targets, and they hadn't been doing the sequencing of all their rounds of selection because, well, that's expensive, or that's what other people do. But actually, guess what? You can learn a lot from what passed and what didn't pass some filter, other than what clone you pick out. So here's the thing I meant to say at the beginning, which is a little bit obvious, but also pretend it's deep, okay? Which is, I just can't get away from it. I have to try to know and understand and develop the methods that will really change biological discovery and bioengineering discovery, at the same time as really knowing the domains and what the technologies are around that people can do. Because if you don't know the domain enough, and you don't know that. So that's the one thing. Although you should go and experiment, you have to know that you'll need to know at least 2 domains. Okay, oh, what was that? That was what I show every cartoon of a protein sequence alignment across some fake phylogeny with bad pictures. And it's just like 5 residues, and you're meant to notice the pink. Okay, but sequences don't look like that anymore. They don't even look like this. This is just one protein across much of life. It's an enzyme called dihydrofolate reductase. It's in all organisms, and I think only about 100,000 are on that slide. So, you know, our brains, however good we are at pattern recognition, which we learn when we're 2 or 3 or 1 or whatever, we're not, we don't really have, there are possibilities to learn what's in there beyond what people thought they could learn 40 years ago. I suppose that was my premise. So 40, 50 years ago, people did, and still do, and very successfully, I should say, is you take a sequence, you can get similar sequences that do similar things by using standard software in molecular biology, and you can look down the column of where it aligns, and you can say, oh, I can't have this thing there, this variable, I have to have a different variable. And as Francis described this morning, the problem with that is that very good try, department, but actually, you know, this residue here interacts with this residue here, right? And it's affected by it. So we live in a 3D world. We are 3D animals. We are interactive animals, and also bacteria interactive, just saying. So in fact, the idea that it was going to be enough to just align these, powerful though it was, when people first looked at myoglobin and hemoglobin and saw where the heme site was from looking at it, wasn't enough. So people then tried to look at pairwise things. So I'm going back in history just to show off, and then I'll get over that quickly, okay? Promise. So people tried to look back, and this was in the 90s, and say, okay, well, you know, if there are dependencies, let's look at pairwise correlations, okay? And so you can use covariance, mutual information, if you want to normalize. And it turned out that people tried that for about 12, 15 years, unsuccessfully. Because what they were trying to do, well, they made a virtue out of being unsuccessful. What they were trying to do was say things that correlate might be close in the 3D structure. Reasonable hypothesis, right? Things that correlate over evolution, they're constrained to be near each other, even if they're longer distance in linear polypeptide. And it turned out that they couldn't get it to work. And they couldn't get it to work because their algorithm was crap, basically. Not crap, but it was mutual information. And what various people did in the late 90s and early 2000s, unbeknown to me actually, in about 2008, was say, you know, this reminds me of Ising models and Potts models and statistical inference. And, you know, there's all this transitivity. It's called super additive correlation. So you get all this transitive information if you just do plain old correlations. And it turns out if you use the right algorithm, so that's the show off bit over, that you actually do get the top ones relating to the 3D structure of the protein enough to early on fold. Just so you know, they weren't brilliant, but the membrane proteins were spectacular. And thereby we found out that actually, you know, evolutionary information has all this information about 3D structure in it that had not been mined in the right way. And there were predictions from that that then got solved. Okay, enough with that. So at the time, we went on to show that, well, if you could do it within one protein, we can do it within protein interactions. So we went genome-wide, proteome-wide. Can we not only say which protein interacts with which other protein, but can we also say where they interact and which positions are important in that? And the rest is history, as they say. Okay. So the genius of AlphaFold was, I think probably threefold actually. One was the scaling of the engineering and the attention to detail, domain knowledge, and combining what we already knew we could do with what we called homology modeling. You've got this protein over here, it's in a rat, it's only got 3 residues different, it exists in a human, it's probably okay, it's probably the same structure. Good, try, department, great, let's put that in. And coevolution for unknown. And really, it was pretty spectacular what they did and what they provided. So what we realized early on, as most people in most fields do, have already known by then, because I was already then 2012, 13, actually, why are we just looking at pairs? Is that because there's only pairs in evolution? There's no other like dependencies, like three-way. Oh my God, if you have to do three-way, I didn't put up the model slides, but can you imagine a Potts model, for those of you who know statistical inference, with the third moment and the fourth moment? Forget it. The amount of data you'd need is like coming out forget it. It doesn't exist in the universe. So walk in the door, deep networks, which I was allergic to, actually, because I think, oh, shit, black box. I just want to know everything that works and how it works, because the great thing about the couplings, and that one was its interpretability, right? Oh my God, I've got to do this thing. I'm going to put these sequences in. It's going to learn all the dependencies, and is it going to be any better? And fast forward, it turns out a little bit for various things, and we can talk about that. So the first thing that we did is work on something like a variational autoencoder, because we read the Maxwelling and Resendi paper, which talked about the elbow, for those of you who know what that is, and variational inference. And we said, this is what we could use for protein sequences. Let's try it out. And it turned out to be pretty good, slightly better than what we'd done before with couplings, which maybe goes to prove that there are higher order dependencies or not. But and then fast forward, there's a very bad cartoon there. I know you're not meant to apologize in the slide, I think, but on the right, which is just showing autoregression. But there was WaveNet. Who knows WaveNet? Dilated CNNs. Woohoo! I tried LSTMs. I came in this business late, but it was 2015. I tried WaveNet 616, which is pretty good, actually, because it's a way of doing faster CNNs just by dilating over what you feed into the next layer. And it did extremely well for generating libraries. And then transformers, oh my gosh, attention, of course. So, of course, we use them like everybody else does, and have developed them now to do something different, which I'll come into later. Okay. What's happened to the, okay, I've already said this. No one model fits all. This is super important for our field, because there's this image of we just have to have the foundation models over here, and then the domain knowledge, which is fine-tuning over here. And you want to talk to me later about that. I think there's an awful lot to gain by knowing the domain initially and doing the modeling with the right representation and calling for the right data. And actually doing the structure of the model differently according to what the data is and what you want to find out. Back to, I'm taking too long here. How long have I had? I've only got 5 minutes. I haven't even started the talk yet. Okay. So this is just a resource, so I'm not going to tell you about it. But one of the things that we found, which I hate as I've gone along in the lab, because I think, why didn't somebody else do this? Which is, I have to produce a resource for the world. I'm responsible for how people evaluate what they do. And they're evaluating it on 3 data sets that they chose because it works. And so we have put together millions of these experiments where people have predicted the effect of mutations by some high throughput experiments, some of them lower throughput, and measured it in different assays, often coupled to sequencing. And not only do we put the data together, but we actually run, I think it's 75, there might be 40 on here. Yeah, 75 models. We're going to cut that down because models are coming out every week, because students and all of us work in a silo of what is defined already as a problem. So it's a good idea to zoom back out a bit. But we run them all, and then you can look at what the results are. Okay. So what did we do? We use the variational autoencoder. We actually, at the same time, in fact, one of the major authors on this paper had been involved in Attention is All You Need, the first transformer paper. But we tried and we tried to get better results with the transformer, and we couldn't. So we had to use this variational autoencoder, never mind defining the families with an alignment. Big disadvantage of an alignment in biology is it has massive assumptions about how you align. And actually, also, it can't actually deal with, generating sequences of different lengths. Not that most people talk about that, but or even, assessing sequences in natural world that have got 3 or 5 insertions or deletions. So we knew it was a hang-up, but it was much better. So what did we show with that? I'm going to try and cut to the chase here quickly, otherwise it's going to get kicked off. So the first paper we did on this, which is about 2021 with the VAE, which was to show, instead of saying, here's experiments that have done mutations and predicted this stuff, where they say, is the protein damaged by some assay, or does it bind this, or whatever it is that they care about. We said, given that the goal of the people who are doing those experiments is to say something about human genetic variation, let's now compare what they do as an experimental with the goal, which is, say, clinical evaluation, with what computation can do with clinical evaluation. And, you know, surprisingly, this isn't to show that computation is better. It's to show you don't need the experiment. You just way don't need the experiment. And this was a huge thing to get through, as Francis described, the snooty journals, but it did. And it's now being really taken up, especially in industry, actually. Okay. Oh, God, but it turned out that wasn't good enough either. That's what comes of being a bit self-critical. So if you take each protein at a time and you say, I'm going to mutate this position and I want to know how bad or good it is for something that I care about, some function it has, you can have a model that has, you know, you have a readout from the probabilistic model of the log ratio of, you know, the energetics or whatever you want to have, whatever you want to call it. And then you can compare it to the experiment. And then you do another protein and you can do the same. And it turned out, if you do all the proteins in the human genome, they've all got the same range of scores. But we know that some proteins, if damaged, if mutated, are worse for you than others. So how naive is that all these guys, including alpha nonsense, I didn't say that, I hope this isn't being recorded, do not appreciate that that is a problem, that we have to be able to calibrate the score across the proteins, because walk in the door with your genome, I want to be able to say, this variant might be bad in this protein. This variant might be really bad for the protein on planet X, but actually for you, it's not going to make any difference. You can alter anything in that. And the original methods couldn't do that. So we combined. So, okay, so how would you do that? Heuristically, you might say, I'm going to combine information about deep evolution, which we were learning about what's allowed in a protein, with population information about, which sort of tells you what is, you know, how much is this protein is part of a gene changed in the population, gives you a clue of how important it is to that organism. So you combine the information, and at the end of the story, of course, is that it's wonderful, otherwise I wouldn't be standing here. Actually, I probably would, I'll probably tell you the truth, but because I can tell you other things that didn't work. But we then combine deep and shallow evolution, which I think is a very interesting concept. You have to think about that offline, how you do that. We just did it with a deep model, a sort of VAE, or you can use a large language model, doesn't matter, based on the protein universe, combined with population information at a very, very, very low granularity, so you don't get population bias, which turns out to be important to geneticists. How did we prove it was right? We showed that we could identify what was already known in candidate cohorts without the use of cohorts, without needing the cohort in the first place. We could find the needle in the haystack. We found hundreds of new candidate genetic variants. And what's worse, including our own, I mean, here, actually, we've got alpha. It's really bad, this slide, okay? Because we should have Eve as well as Pop Eve, because that's our earlier one, right? Because our Eve was only a bit above alpha makes sense. But what we found clearly was that if any of the methods that exist, and there's a million of these methods, it looks like a saturated field, but it's so not. In terms of anything sensible. we found that it massively over-predicted. Imagine you said everything was pathogenic that you looked at. Then you'd get 100% recall. And basically, that's what they were doing, statistically slightly different to that. And if you compare what they were able to recall with severe developmental disorders and how many of those extremely serious, immediately phenotype labeled at birth, everybody would have like 100 of them, according to AlphaMedSense or Revel or whatever else anybody used. So a very important, and this relates to a very important point, and relates to the fact that in machine learning and AI, we need to develop, especially in biology, I think maybe it's more important than other fields, I don't know, you tell me, where the way that we evaluate it has to directly relate to what we say we can do. So otherwise, okay, finished. The main thing there was, it turned out, this is what all the geneticists are interested in, that we don't need parental genomes. Because they go out and they have to get trios, they have to get the parents and the children. It turns out we don't need it. We can find the needle in the haystack, but the score tells you something about the severity. Okay. And just in case you think, oh, I still haven't done the virus. Oh, shoot. Can I have like 2 minutes? 2 minutes. Okay. You heard a bit about a virus this morning. I have to tell you this because it's cool. Okay. So it's the kind of other end. So you've got a distribution of scores that you learn from evolution, and you've got the really stuff that says it's bad. And then you've got the stuff that says, this is great. If you want to change this, it's fine, at the other end of the distribution. And so it turns out that if you, let's scoot ahead. If you take sequences of things that change in viruses over time, especially things that people are concerned about, vaccines for flu or Ebola or Lassa or, I can't remember the name of those viruses, Chang. Vanilla. Okay, you have to tell me. And it turns out that there's a huge corpus of information that people haven't really looked at. And so can we learn what's mutable? And it could be that viruses, right, change. So there's so many possible landscapes that they could go to, that why on earth would the history of that sequence tell us where it is going to go to? But guess what? You can do an experiment on a computer to find out if that's true, because we have the pandemic to so this is just what the model is. The model had an AI evolutionary sequence model, which you can put anyone into it, actually, and then a couple of other things which you may or may not need to say, okay, if I want to predict how this bit of a virus evolves, what models would I need on what, and what do I need to consider? So that's just to tell you the models do, we did that. That's to tell you how experiments didn't work, but I haven't got time. Okay. So I'll finish with this, I promise. So I said to the lab, we're not doing virus, it's pandemic. I know you all want to have a go, but we're not doing it because it's too opportunistic. But we ran some models and we said, you know, what we noticed is if you run models on sequences pre-pandemic, no SARS-2, okay, you can say something about what then happened to SARS-2. But everybody said, well, you can't do that. You must be cheating. No, you're not cheating. You show that you're not. And it's something to do with the fundamental biology. It's not our cleverness of our models. Our models are just learning the dependencies. And they're saying that in the history of this interaction with our host is written in the sequence something about the interaction with our bodies or animal bodies that then can be learned. So how do we evaluate this? So, okay, I'll do this and then I promise to stop. Okay, are you all bored? Okay, you didn't say no. So the blue line is a time on the bottom, and it took about 9 months before anybody got any antibodies and tested them. And they test them against, say, all single mutations, because you can do these high throughput experiments now. And that's what the green line is, what they would have predicted compared to what happened. And then later experiments, I think 1 year later, they get a bit better. It turns out that the blue that you could have done on January 2020, you get even better. No data from post-pandemic used whatsoever. Kind of crazy, right? Hard to convince reviewers, but it's there. And now, fast forward, we have some implementation and further validation. Okay. Actually, that's just the flu one. Not only can you predict better which ones are going to appear in viral evolution, you can also predict the ones that are the most frequent. Okay, we've done it for flu, we've done it for 1 million viruses, and you can find all the other things people care about. And you can design things and check and test them. And on this slide, what we show is that the ultimate thing, which is what we want to know, which is a big challenge that's unknown in biology, it's not really known in public, as it were, is how can we map what we see as human beings or an animal sees with our immune response? And it's not, you can't just do it by similarity of sequence. You have to learn something else. And the triangles are things we designed that were not like the circles. The circles were real. The triangles are what we designed that were totally different and go to the same immune space. So now we're designing vaccines. Wish us luck. I'm not going to say anything else. I didn't say enough about co-designing computation experiments. So I'm just going to say that in yellow now.
Thank you
Done.
so much, Debora. This is just absolutely fascinating, and I'm sorry we couldn't hear all of it.
Well,
I hope you'd be willing to share your slides with everyone. Yeah.
And one day I'll do, a short talk.
Are there any quick questions? Okay, well, we will all be joining you.
Yeah, there's one there.
Oh, great.
Oh, but it's Chang, it's product placement.
Hi, Debbie. So in the missense prediction, can you explain a little bit more of why the scores are all similar if you just look at each protein? And then also, to what extent do you think most of the scores are driven by stability effects?
Two different questions. Let's do number one. Number one is, why might it be that there is that artifact, which actually, we want to revolutionize human genetics. Come on, we can read and write human genomes. We can't interpret them. What is wrong with us? 23andMe, she's in jail or whatever. Is it her? Anyway, what I mean is that, what's wrong with us? We have all these other goals and we can't even do that. Why not? Okay, so one of the things there is evolution is just a powerful, a very powerful record of billions of experiments, but they are each of their own, the proteins in different evolutionary histories. And what they do in us is at best is a secondary effect. It's probably an equal effect, which is how important it is for us to be able to walk up a mountain or do code or talk to each other. So there are organism-specific constraints that are not captured by deep evolution that we can capture. You can imagine capturing that by using a heuristic like what is fatal in a mouse if you knock it out. But this is a more machine learning method where you just use the population information instead. And I think the reason is because, actually the fundamental reason is because the evolutionary information is very, however good the models are, is just very sparse little dots in the universe, right? So, didn't answer it. Second question I haven't answered. Oh, stability. Oh, everybody says that. Yeah. Primary thing is stability. Okay, fine. And what's everybody interested in? What this protein does in a body, like what it interacts with. Yes, it has to fold, but it's not like fold or bind this or do that. It's one of them is a higher order thing, and of course you can do that with.
Well, it just seems like stability is something that is more consistent across.
Yeah, exactly. Well, to some extent, although you have a distribution, because it's stable enough, isn't it? Not stable. Okay, thank you.
So much. And we will all join Deborah at the Huntington Gardens, where we have dinner and a reception and some short talks by a variety of Caltech faculty. So when I first saw this on the schedule, I thought it was a real Southern California flex to have us go on a 30-minute walk on the 10th of November, but they actually very kindly arranged buses for us. So there are 2 buses right outside the doors ready to leave pretty much immediately. Oh, yes? Oh, okay. So please follow this gentleman in the back. He will take us to the buses, and they will take us to the Huntington Gardens for dinner and a reception. I'll join you all there.